swollen couple of cells, it may not be that harmful. So let's look at a couple. So a tumor, if it is benign, then it won't spread. This tumor, which is benign, it will be a non-cancerous. So when I use the word cancerous, that means it will not, it has to spread. And if it's non-cancerous, then it will not spread. It is not able to break certain kind of membranes that is holding it. Now in this case, it's a circle, but it's not always a circle. It can simply be a lining, can be simply be an extra membrane here. And so they are not able to spread. They are really contained. But a malignant one is able to spread. It is able to break through this membrane, and it just can make certain conditions that it is able to make and break through the membrane, and that is cancerous. So a malignant tumor is cancerous. So anything that's benign might be a tumor, but it may not be necessarily cancerous. So if it is benign, then it will have the suffix of oma, so adenoma. Okay, so it will end with an oma. That means it's contained. It's not so serious right now, and it can be taken care of quickly. But if it is a malignant tumor and it has the capability to spread, then it will have the name of carcinoma, or it will have the name of a sarcoma. So such as the name is adenocarcinoma. So let's look at how do we name these cancers? How do we, where do the cancers come from? So how do we classify? So its classification of these cancers is based on the tissue of origin. What is the tissue of origin? Where is it? Where did it start? And so which is the starting location. So if you see here, there are four types: epithelial tissue, connective tissue and muscle, brain tissue, and hematological. So epithelial tissue, that is, and we'll look at each of them in a tad bit more detail. So epithelial tissues, if the cancer starts from epithelial, which is the most of the cancers, then it can be either benign and it can be either malignant. So it would be called as a papilloma or an adenoma, or it could be a carcinoma. So just to give you an overview before we get into the details. And so if it is in a muscle tissue, muscle means it could be connective, like bone, cartilage. Then in benign, they're contained. They're not very serious. They can still be fixed. They can still be treated. Some of them are rare, some of them are common. So fibroma, osteoma, rhabdomyoma. But if it becomes malignant, then it is called as a sarcoma. So sarcoma is the name given to connective tissue and muscle cancers. The word glial refers to brain. So glial cells, and I'll show you a diagram later on, that they give rise to brain cancer. So brain cancers will be glioma, meningeal sarcoma, or meningioma if it is not very, if it's just benign, and retina, and I'll talk about retina and retinoblastoma. So these are brain, central nervous system related. The other one is blood and lymphatic system, blood cancers, leukemia, lymphoma. These are all blood cancers, so hematological cancers. But lymphoma is from the lymphatic system, and these are leukemia are in the lymphocytes in the blood. So they can start in any one of these, but then they can move on. So epithelial are the major ones. So glandular and ductal epithelium, this means that it is the coverings, this surface one is the covering of the organs, and these are for ducts. Ducts, like your cells in the intestinal lining, they have to secrete enzymes. So they will have duct to secrete enzymes. So let's look at that in slightly more detail. So epithelial cancers, they make up the majority. All right. So about 80% of all cancers are all epithelial. So they're all epithelial cancers. And what is epithelial? They are on the surface of the liver, surface of all the organs. It's just on the surface of all the organs. And these will be, solid malignancies. And the hematological malignancies will all be blood related, and so those will be here. And so some of them are also yeah, they're the ones that are down here. These are the only ones. So let me just go to the next one. This is the same. Hmm. So when we do the cancer types in the next talk, when I'm doing the cancer types, that's where you will have more details on the individual cancer types, which is the names that I gave you, the sarcoma or the adenocarcinoma, etc. So we will look at all the details of the individual names. But right now, so far, you understand that they are based on if they are carcinoma or adenocarcinoma or sarcoma or a myeloma or leukemia lymphoma, then these are the ones that are most scary. These are the ones that are the more deadly ones. So those are the malignancies. Okay. So that we will save for our next talk. So the next one, I just need to know how much time I have. Um, Sorry. So what is the process? How does it become a normal cell, become a cancer cell? What gives rise to this primary tumor to go to other places and become tumors in other places? What are those hallmark capabilities? This is a very special word I would like you to pay attention to. Hallmark capabilities. What are the hallmark? That means those have to be those fixed properties. And so if a cell has those hallmark properties of cancer, then how does it work? Is it a multi-step process? So let's look at this. And so in order to understand this, you really need to understand something about the um, risk factors. So something a little bit about the risk factors need to be understood in order to understand the full mechanism about why the hallmarks are there. So these are risk factors. That means they result from, these kind of risk factors, about 9 of them here, that lead to either result in genetic mutations and they drive transformation. So please note this very special word. The word is transformation. And the word transformation means that the cell has been transformed into a kind of cell that is abnormal. So transformation is a very specific word. So in a normal cell to become cancer cell, in mammalian cells, transformation is a very specific meaning. So and so these can always be tobacco, microbes, bacteria, obesity, family history, and so all of these together. And so some of these are, alcohol, your occupational exposure, exposure to asbestos, etc., and a poor diet, okay, poor diet, obesity, etc. These are that are known. And so what happens is that when these risk factors, the burden of these risk factors give rise to that genetic mutation. And so what will happen is that some of these cells that need to die, generally these cells will damage and will die. They will just not die anymore because they have acquired that genetic mutation, and they don't have to die anymore. So generally, if a cell has to, normally a cell will die by suicide. It's very, very normal that a cell will die by suicide, but in this situation, in a cancerous cell, they will acquire a mutation. So the first mutation, then there might be some of them, not all of them, will acquire a second mutation. Some of them will acquire a third mutation. And a mutation is a change in its DNA. And a fourth or a later mutation, and then that some of those. So not every cell from here, all four, but maybe one or two cells the cells are really, disorganized looking, and they are pretty much everywhere. And that is a cancerous cell compared to the within the normal tissue, and so it's extending and moving forward. So, are we okay, everyone, so far? Pooja, are you all able to hear me okay? I don't see anyone. I can't hear anyone. So now let's talk about a little bit about the um, cervical cancer, which we like to focus on a bit more in certain locations, and you will do also your workshop has a little bit focus on that. Oops.
Hello,
Yes,
ma'am.
yes.
Yes, Pooja. Pooja, is everything okay? Can you all hear me okay? Can you see me okay?
Yes, ma'am. Everything is okay, ma'am.
Okay, all right. So, so the example of, so we looked at a couple of examples, and cytology is what we're trying to understand a bit more here. So in the cervical cancer, this is the cervix, this is the female cervix, and the cervical cancer is a preventable cancer. Okay, we will look at that again in the next talk, but cervical cancer is a preventable cancer. It takes long, many years to develop, but if it is can be screened early on, then it won't develop and it will not give rise to the deaths. And we will look at some of the data later on. So this is a just as a normal. So if you can see that this is the normal visualization of the cells. This is the tissue, right? So these are just tissue. If I were to take a biopsy here, all right, this is just the tissue. But first of all, these, in order to, if there was a infection of a virus, the HP virus, then they can undergo these changes, but it takes a while. And so the first changes is abnormality, and that is called as dysplasia. So it is called as dysplasia. I hope you can see my pointer. And those are just abnormality. They're not cancerous yet. They're like pre-cancer. And so they are normal cells. So this is your normal tissue where you can see that the cells, there are two different boundaries. These are your cervical cells. And so when there is some kind of a mild dysplasia, right? So there's a mild dysplasia, then it's called as low-grade intraepithelial lesion, LSIL. It used to be called as a different terminology before, but in the last couple of years now, this is the major terminology used. So a mild dysplasia means mild changes. How do you know the changes are mild? Well, you can just see in this situation, in this one, every cancer is a bit different. So in this situation, the cells have, the nucleus have gotten bigger. So you can see that the nucleus have gotten a little bit bigger. They don't have their normal stratification, as you can see here. See that nice stratification? They don't have that. They are just like a disorganized mess, or slow going there. Then there is a little bit of like a high grade or moderate dysplasia. So it could be moderate or severe dysplasia, in which, in this particular situation, the nucleus has become fairly large, and each of these cells just has a huge nucleus, and that's a very, very unique characteristic for cervical cancer. And the last stages are, when it becomes cancerous, really, is when it breaks open this basement membrane and it starts to extend out. These cells, they start to develop mutations such that they can now go and extend into the other locations. And so, therefore, if we can stop it at the early stage, sorry, then we can prevent the cancer from taking place. So, dysplasia goes from mild to severe. So the cell has to acquire certain capabilities. And so that's the word. The cell has to acquire certain capabilities. These are called as hallmark capabilities. Hallmark is, if I don't see them, then it's not cancer, but those have to be met in order for the cell to become neoplasm. Neoplasm is like an abnormal cell, you know. Um, so normal cells will start to transform and become a neoplastic, something different, something abnormal before it becomes cancerous. So those hallmark capabilities, I put in, 6 hallmark capabilities for now, about just to so that you understand not so much of molecular biology here, but that these are phases. These are phases, or these are properties that the cell has to achieve in order to become cancerous. And so if you start from number one here. So on this side, these are the 6 major ones. Sustaining proliferating signaling. Signals are small molecules, small chemicals that one cell secretes and tells the other to follow. All right, so one cell secretes it, and the signal goes to another cell, and then the cell says, "Okay, I got the signal. I will now, you know, cell divide." Sorry. And so if there's too much of signaling for proliferation, then that becomes a problem. So cancer cells will continuously signal to keep dividing. So one cancer cell is telling another cell to keep on dividing, and those signals just do not stop. Those signals, the cells will take up the signal, and then they will continuously divide. So those signals are not supposed to be there all the time. So sustained proliferative signaling. Then the next thing is that, so too much growth. Then how about inhibition? Cells are supposed to have some kind of suppressors, so growth suppressors. And so those growth suppressors, the cell will just not listen to them. So these are important growth suppressors here. Their name is RB and P53. These are tumor suppressors, and they stop the proliferation, but nobody's listening to them. So the cell is going to develop mechanisms, methods to stop listening to the growth suppressor. So evading the growth suppressors, important master molecules, RB and P53. Cell refuses to listen to them. The third one is resisting cell death. Well, if a cell becomes abnormal, normally on a day-to-day basis, the cell needs to die. It needs to die. The cell will evade cell death. And that is called as apoptosis. Apoptosis is a cell death, a very specific, very programmed cell death. And so other programmed death pathways that are supposed to be followed, the cell will find a way to avoid the apoptosis. It will shut down any apoptosis pathways. It has proteins that will just ignore that. Then the cell will achieve immortality. Cell will achieve immortality. This cancer cell will now slowly start to do things that will achieve immortality. How will it do that? In its chromosomes, it will install some telomeres in its chromosomes. Telomeres are ends, the end of the chromosomes, and it will keep on installing more telomeres so that it never kind of gets old. It keeps on dividing. So it becomes immortal. Every cell has this job to die at some point. And this one will now even surpass that. Not only will it not die, but it'll say, okay, I can keep on multiplying. So it will achieve immortality. Not in a good way, of course. And then the cell will say that going to make a tumor, so I need to have blood vessels. So if a cell needs to have blood vessels, then it's going to induce angiogenesis. Angiogenesis means blood vessel, making blood vessels. And making blood vessel is not, for all cells. And cancer cells will develop and learn how to make blood vessels and give the signals. So it will stimulate new blood vessels so that it supplies, blood to the tumor. And the sixth one is that it will learn how to travel. It will learn how to be motile. The cell will learn how to use its own proteins to be able to crawl, literally crawl and go places. And so it will then evade. Sorry, again. I don't know why this is happening. So, and it will invade and go to distant sites. So it will activate invasion. Invasion is going across the barrier. So guess which is invasion here in this one. Invasion is here, where it is crossing the barrier. from one type to the connective tissue. So from here to connective tissue. So it will invade and then it will go into the blood vessel and it will go to distant sites, and that is called as metastasis. So metastasis is going to a faraway site. So these are your six hallmarks. And in all of this, somebody has to support these tumor cells. So if somebody has to support this tumor cells, who's that going to be? It's going to be the neighbors, the microenvironment of the tumor. Okay, we're not going to talk about this too much right now because we have a no whole separate lecture for this. So the microenvironment of the tumor is a bunch of cells that help it to progress, that help it to go to the later stages. So it supports the cell transformation. And so these are all blood vessel cells from the blood vessels, etc., to give, oxygen, to give blood. But the immune cells, really, it is the immune cells. So the microenvironment has all these immune cells, it has blood cells, etc., nearby everybody, every other cell that is going to help this tumor to grow. And so we can target those immune cells and tell them not to support the tumor, then that is called as immunotherapy. So your immune cells, your lymphocytes, they will have to be told not to help the tumor to grow. So that is for the next talk in lecture 4. What is the cell going to do? The cell is going to change gene expression. Now, we're not going to talk about gene expression in this particular coursework, but just to let you know that the cells will modulate their genes and tell the genes to do more or less of something, and therefore they will turn their genes to activate the blood vessels. And so for all of this, there has to be multiple steps. Many steps need to be there. And if we combine all of those multiple steps and add in further requirements, then we find out that throughout these multiple steps, all the multiple steps for all of these, they are, and including the tumor microenvironment, we now, it seems to have additional hallmarks, additional requirements, hallmark 7, 8, and 9. And so if you look at 7, 8, and 9, besides what we just talked about, besides what we just said, you have, proliferation, evading growth. We just talked about these in metastasis. 7, 8 is reprogramming cellular metabolism. These three, 7, 8, 9, are a little bit new. And so reprogramming cell metabolism means that um, the tumor wants to know, should I take glucose? Should I not take glucose? I have a lot of glucose. Maybe I don't want the glucose. And should I take more energy? What should I do? So reprogramming that. How can I be more efficient and be a bad tumor that way? And number 8 is avoiding immune destruction, means all your immune cells, they have a job to do. They're supposed to go and um, uh, to make those cancer cells die. But the immune cells suddenly will actually start supporting the tumor cells. So they will avoid immune destruction. And lastly is that they will undergo major changes in their own differentiation. They will acquire properties such that they can stop other cells from doing what their job is. And so they will be disruptive, and they will acquire these various properties. And so here in this picture, you see that there is blood vessels growing around the tumor. See that? These are these blood vessels. These are these blood vessels going around the tumor here. And the some of the tumor cells, they can enter into the blood vessel, and then they can go into other locations. And so this is a tumor that is being fed by blood vessels, and it is going to also later on metastasize, and it's going to go into different directions. And so those were the three ones were really these new hallmarks. Okay, so these are called as the emerging hallmarks. So 7, 8, 9 are emerging hallmarks. That is new. And so there's a lot of things for you to understand. So increased glycolysis. Well, uh, will they break down the sugar or will they not break down the sugar? So these two I just said are avoiding immunodestruction. And how again? How are they doing this? So I started out with six, and then I had 7, 8, 9. How are they doing this? Well, enabling characteristics means that the cancer cell is like very, very, it's kind of like getting smarter and smarter, and all it's doing is it's undergoing certain mutations, and it's going to evolve. So increased mutation rate, they're going to have a mutation, even more mutation, that fuels its evolution. So it's getting to become a smarter, um, in a bad way, a smarter cell that is going to have, all because of more mutations and genome instability, it will acquire all of those hallmark properties. And the tumor microenvironment, like the surrounding cells, the lymphocytes, they are going to help it. And so those are the enabling characteristics. So because of the tumor microenvironment, 7, 8, and 9 can take place. So I will kind of like now quite, wind down this talk. So, how is this relevant to you as computer science and IT students? How is this relevant to some of you? Well, right now, like I said, we are not going to focus on proteins, but maybe later on, I just want to keep you in that loop. So, all the hallmarks, this is what your job is. All the hallmarks, like its proliferation, having blood vessels, not dying, okay, evading death, and signaling pathways. These are all the different kinds of proteins that you can stain the cells and look at them. These are all signaling pathways that get activated. So those can be, one can examine them. So you can look at these pathways, you can examine these signaling networks. So you can do signaling network modeling. You can look for all those pathways. There are many pathways. You can, I'm not even saying visualization of cells, that's more straightforward, but you can look at all of these pathways. So each of these hallmarks that I just described, they have certain proteins. Everybody has a specific bunch of proteins that enables this. And so maybe you can map them and you can make a circuit of them and you can analyze them further and see if they are talking to each other. Maybe P53 and growth factors, how are they all integrated? Maybe VGF for angiogenesis, for vascularity, and P53. Are they doing anything together? Is one after the other, or are they working together? These are the things that you can also bring up. So signaling network pathways, and then from there you can look for drugs. And so a lot of the new platforms, etc., they all utilize that kind of expertise that you can provide. But of course, for this particular, let's go all the way down here, look down here. For this particular Gyan program coursework, we are really looking for trying to look at image analysis for these cells. So we are trying to look at morphology and tissue-based stuff. So there is more things that you can do. So overall, your carcinogenesis, what does carcinogenesis mean? That means how did the cancer develop? What did you just learn today? That number one is that there have to be some very, very specific mutations and they keep on acquiring. Then that there are hallmarks of cancer, that properties and gene expression that are taken. So those hallmarks are acquired also by gene expression, by gene alterations. And then you looked at some cytology and some basic morphology of cells, of differences with normal cells. And so with that, I will end this particular talk. I hope I have been able to um, do it in time, such that we have some time for question and answers. And so our next topic, after a little bit of a break, will be to go into a little bit more detail and understanding the cancer types and the names that I have not talked about, and and look at what the world, how much cancer there is in the world. So,
Thank
um,
you, ma'am.
yes, please. So, if there's any questions, I will take it here.
Uh, I I want to that is from the participants. Uh, Please ask not any question. That is the how to use or as a distinguished person, Professor Nath. You can use the ideas in our programming. That is not that is the new ideas, innovations. We can use it. So I think the students, the research scholars may interact. No issue. That is the every time only the that is the any complex question. This kind of not a issue. I think. So again, Somudeep, are you present here? Somudeep, acha. That is anyone that is Professor Deepak
Yes,
and
yeah, we have time. We have time. They can surely ask questions. Can I pause recording for a bit now, or should I continue the recording?
Yes, ma'am. If there is no question, then we can take a break for 15 minutes.
Yeah, yeah, and we can have questions, when we come back, and we can have questions even right now. Um, I don't know, am I following time? Because I'm looking at all the wrong times.
No, no, ma'am. It is right.
It is on time.
11:15.
Okay, okay, thank you.
Ma'am, then ma'am and everybody,
Recording stopped.
uh, we will be there on 11:30 a.m. for the lecture 2. So we have 15 minutes
Yes,
break.
so the way I would request everybody to do is you can just stay on this Zoom or you can come back in a bit. And I will just keep this Zoom on. And you can just keep yourself muted and just join again. That's all. It's the same Zoom. And I will be here if anyone needs to speak.
Okay, ma'am. Thank you, ma'am.
from the first year, Debujit, any other our
Yes, sir.
okay. So Debujit and I think Tonmoy, Orno, Debujyoti, Shostika, Priya, Johnny, Priti Ranjan, Ronit. So I am giving the opportunity the younger persons first. So any kind of question or any kind of any that is Devojit, Ronit or no. So you can interact with madam, no issue. Devojit, any question?
No, sir.
Regarding
Right now I don't have any question.
okay. So here that is in the cellular level, how can we uh, how we can detect the different machine learning methods like segmentation, detection, classification, this kind of thing with a prior knowledge of the biology and
So,
how to use.
Dr.
Okay,
Bhomik, let us, go through one or two talks, and then today you will get a better understanding of what,
sure.
what other interests they have. Yes. So I will try to give as much as cell biology from my side as possible, more on cytology and less on proteins, but there is going to be one talk where they can look at proteins. And we are doing this, so we do need, computer science students to think about this. So cytology being important, but yes, further. So let us go through at least one, two, three talks today and see how it all works out. And I think that they will start having questions. Right now,
Sure, sure.
this is the basics.